First of all, I would like to thank FEBS for organizing this informative workshop and the Youth Travel Fund for funding my accommodation and travel. So today uh, I would share with you our current knowledge on prokaryotic KU, or in specific mycobacterial KU, and its role in double-stranded break repair. So before I begin, I would like to give a brief introduction of what a double-strand break is and what are its fates. So a DSB can be introduced into the genome by ionizing radiations or endogenous factors such as ROS or reactive nitrogen species. So once introduced, a single DSB is sufficient to kill a cell. For that, cell has two specific mechanisms. One is template dependent, where there is a need of an extra copy of the genome to repair this damage, such as homologous recombination. And the other is template independent or non-homologous enjoining, where there is no need. So the template dependent mechanism only works in the actively replicating cells. But in case of non-replicating cells, as there is no extra copy, the NHEJ is the only reparation method which can be used. So which is more specific in case of Mycobacterium tuberculosis, which resides within the host macrophages as dormant cells, and they're constantly bombarded by genotoxic agents that cause these DSBs constantly. So for that, they require a robust and fast DNA repair mechanism such as NHEJ, which brings me to the prokaryotic two-component system. So there are only two proteins involved here. Number one, that's Ku, which binds to the DNA and houses the component 2, which ligates the two ends. So, but there are two critical factors. Number one, what senses this damage and triggers the response. And the second is, or more important, rate limiting step, that what brings the broken ends at close proximity. If you look at the human NHEJ complex, it's complicated with n number of proteins creating this synaptic complex. But in case of bacteria, it only has these two components. And we ask this question, then what brings the ends together? So ligase D cannot do it because the Ku is the first protein that comes and binds to the DNA, and it should form the synapse. And that was our initial hypothesis. Then we moved on and solved the cryo-EM structure with the DNA as a super complex. I should tell the protein is 32 kilodalton protein, but we were lucky that it could form higher order oligomeric complex by repetitive formation of adjacent dimers one after another, and the DNA is a 40 base pair DNA here. The talk that I'm going to give now would be based on several cryo-EM structures that we solved, and the HDX-MS data, and few FRED-based assays, and also some biochemical and biophysical assays, which are present in this research article. So the question that I would ask is what are the early events of prokaryotic DSB? How the unique DNA synapses is formed in prokaryotes and what its architecture is. And third, I would like to show how we can capture the dynamics in live cell. So if we move on, first, let's have a double stranded break here. And as you would imagine that these ends are highly flexible, and more so it's prone to nucleases, then it causes the loss of further information. So to avoid that, you have the homodimer, that is the protein Ku. By our experiments, we know that it stabilizes the DNA and protects the ends from exonucleases to chop these ends. So if you look at the protein itself is very interesting because it has a very high thermal stability at 50 degrees in its apo form, and when it's bound to the DNA, it shoots to 70 degrees, remarkable in that sense. And it has partial resistance to SDS-like detergents. So when we looked at the structure, it looks like two human hands clenched together, where the beta turn beta motifs resemble human fingers, and there are two slings which transversely cross this complex and give further compactness to the structures, thereby explaining its stability in solution. Now we wanted to look at how it interacts with the DNA. So here there is a 2D map of the amino acids interacting with the DNA strand, and by in silico alanine scanning, we screened out the critical residues, which may be the critical residues for this interaction. And if you look at this wild type structure, you would see a positively charged pocket perfectly suitable for DNA binding, non-specific DNA binding. But if you mutate them, obviously in silico, you see the loss of that charge at the center. We did MD simulation on the structure as well as the model. The RMSD shoots to double, which tells you that it loses stability and DNA binding affinity. Also, the overall protein gets a little bit distorted, and the DNA sort of slides out from the pocket, explaining the lesser DNA binding affinity. We have EMSA-based and SPR-based experimental evidence to back it up. And then let's go back to our damaged DNA. And when one end of the DNA is bound to the protein, to bring this two ends closer, you need another copy of the same protein to create this synapse. Now, let me take you deeper into the synaptic interface of the protein. When we looked at the synaptic interface, we saw there is only one beta hairpin that is mediating this interaction, specifically residues 12 to 15. So we asked this question, or you might also imagine what would happen if we delete these 4 residues. We did so, and using AlphaFold, we modeled this complex, and it shows destabilization of this synaptic complex as well as flipping of the adjacent protein. We also did experimental validations, which I'll be showing in my next slide. Yes, I'm here only showing the SPR-based results, where you see the wild type that is in green, whereas the DNA binding impaired mutant and the synapses impaired mutants are at red and blue and black as well. So if you look at the kinetic parameters, you would see that the Vmax has been decreased to orders of magnitude, as well as the increase of KD to double or furthermore. In EMSA-based experiments, we either couldn't see any complex forming, or the complexes were a bit smudgy or absent in that case. So that proves that these sort of mutations in this area of synaptic interface and the DNA binding pocket or core does impair the protein of its functions, of 2 main functions. Number one, that the synapse formation, which is residue 12 to 15, and the DNA binding. And these 2 functions are critical for the protein and for NHEJ, because it's the rate limiting step of the repair process. Next, I would like to show how we captured initial dynamics of the protein within the cell. For this, there was a rationale that the protein cannot bind to a circular DNA, and the bacteria has a circular DNA, intact circular DNA. So because of its nut and bolt assembly. So what we did, we created a conjugated construct of the protein with GFP and incorporated it into M. smegmatis cells, a close relative of MTB, whose own NHEJ apparatus was deleted, so it cannot interfere with our experiments. So once created the DNA damage by laser damage at 405 nanometer laser at a region of interest of 1 by 1 micron. So by doing that, we created a site-specific DNA damage, and you would see there is a rapid influx of the protein at this damaged spot. We went on to quantify this, and you will be able to see that there is a photo bleaching event, that is the DNA damage, which responds to the DNA binding dye Hux used here, and simultaneous increase of the MKU GFP signal, as I showed you in the previous video. But you cannot capture the dynamics because it's too fast at that point, and our acquisition time is impossible. We also did raster imaging correlated spectroscopy, but the data, I don't have it here. I can share it with you if you are interested. With this, I hope, doesn't work. With this, sorry. Yeah, I'm almost done. So with this, I hope I could answer the basic questions that I asked in the beginning of the presentation, and I would like to thank both of my institutes and the people I've worked with and learned from, and you for your attention. And I would be happy to answer any questions if you have.